Southern blot. Used to detect DNA. Northern blot. Used to detect RNA. Western blot. Used to detect protein. For easy. Remember. Snowdrop. As mentioned inside slide. These techniques depends upon weight, glass plate, paper towels, Whatman filter papers, nitrocellulose papers, studies gel, saran wrap, support, and tray, as mentioned inside slide. For example, for Southern blot technique, DNA must be extracted and digested by restriction enzymes. Then, detect digested bands by agarose gel electrophoresis. After that, blot agarose gel by blotting buffer to easy use probe. Next, detect blotting gel by expose X-ray film, as mentioned inside slide.